All right. Good morning, everyone. So I know I've been getting a lot of messages concerning when will I post the grades and everything. Hopefully by this week. I just basically the course coordinator has to just let us let's say submit them to you guys. That's it. So but hopefully by this week you will get them. But like I remember I was thinking that you guys were gonna do quite bad because I got a lot of negative reactions during the exam. But honestly, no, the average was quite decent. I think it was in the 70s. So I think you guys did quite well. So hopefully by this week I will submit all the grades. And of course, if anyone has any sort of questions, wants to see where did they go wrong or anything like that, of course, more than welcome to schedule a meeting with me and we can go through them because as you guys remember, the um there's no, let's say, I don't have any more this kind of after class hours anymore. It's just whenever you guys want. So I'm gonna start sharing quickly right now. Can just one of you guys let me know if you guys can see my screen?
Yes, we can look.
Thank you, thank you. Okay, so now we're moving on to chapter 12. For your own, basically for your own information, chapters 35 and 36, which is the plants one and everything like that, I'll of course be sending an email to remind everyone about this, is chapters 35 and 36 because of the current, let's say, war and the delays that we've had, along with now that we have, I think, Friday, Monday, and then the other ones as well next week. We have a lot of these days where we're gonna miss class to keep up and not bring you on a Saturdays and all this kind of stuff. What we're gonna do is that we're gonna make it asynchronous. Chapters 35 and 36. Everything is recorded, everything is there. Please study it. And one of these days I will be having this flipped class and where I'm just gonna, we're gonna come to class and you guys are gonna ask me all the questions about 35 and 36, chapters 35 and 36. So I will send an email just to remind you about this. So we're now going into chapter 12, which is patterns of inheritance. So this is, I think for quite a lot of you guys will be quite a recap because this is much more what you've learned in high school about genetics and all that. So there's always been a mystery when it comes to heredity, and specifically before the 20th century, before we knew what genes were, what all this genetics were about. We didn't know what was happening, but what we were always observing is that why were children looking or resembling, to their siblings or to their parents or to, let's say, their cousins, people that are related to them. Why was that? People really were, let's say, kind of puzzled about that because, of course, makes sense. We didn't know that there existed genes. We didn't know these things. But what we basically observed was that, okay, heredity occurs within a species, meaning that I can pass on my, let's say, characteristics towards basically my son or my daughter, but I cannot pass it on to a horse. I cannot pass it out without outside my species. So this hereditary, this passing on of the traits only occurred within one species. So there wasn't basically being passed on to a horse or a cow or whatever. And we also saw that this was transmitted directly from parent to offspring. Okay? So those were the things that we started to observe. Then came up, we started to basically start to think, okay, could this be actually that we're passing on our traits through fluid? And specifically, we're fluid by fluid, we mean blood. Was it that we're passing on these characteristics through blood so that, let's say, my son would be technically a mix of my blood and my wife's blood? That's what we used to think back in the day. And that was the whole thing. And this is why, if you ever basically anyone is into, let's say, breeding or stuff like that, like animals, there's always been this thing of like, okay, what is the bloodline of this dog or of this horse? Well, this is where it came from because we used to think that traits were passed on through blood, hence bloodlines. And of course, back in the day, and with royalty, there was always this thing about royal bloodlines and stuff like that, meaning that only royal blood would be passed on, things like that. But this makes a bit of a paradox because if this is true, we would basically look all alike at one point because they keep mixing and mixing and mixing and mixing and mixing. It's gonna come to the point where there's gonna be some sort of normality, not normality, but like everyone's gonna be constant because the mixing is gonna end up to that. That's what we used to think because of course, if we're coming on from let's say two human beings, or let's say our population started off with one or two human beings, why didn't we then all came together, like all look like each other? Isn't that a bit of a paradox? Then later on, bit by bit, we started to do experiments to really show that this is not true. It's not really that blood is just being mixed and that's how we're being formed. No. Then it was basically specifically these early on experiments, and the first ones were from Joseph Colruyter in 1760. And what Joseph Colruyter did is he technically crossed different tobacco strains, especially in the 1700s. There was a lot of different tobaccos, plants with different strains. So you got basically hybrids. Hybrids technically means that they're a mix of different traits together. That's what a hybrid is. So it's not, let's say, 100% of one trait, but no, it's a mix of different traits together. So what he did is that he basically crossed them together, and then the children of these two tobacco strains that they crossed were completely different than the parents. But how is that possible? If that if this basically holds true, what they're saying here, that traits are transmitted directly from parents, and it's basically the bloods coming together, then these two hybrids that you crossed, your offspring should look like one of the parents. But no, it didn't. He actually saw that there were some different variations. So this was the early on experiments showing that, hey, this is not maybe true. Then came T.A. Knight in 1823, and he was doing a lot of crosses, not with tobacco strains, but with garden peas. And these are as we also clearly know, these garden peas that we see basically in our garden. They're called Pisum sativa as well. So what he did is he actually got two true breeding strains. What does true breeding mean? So one thing about peas that is quite, sorry, I need to quickly let people in. There you go. One thing that we see technically when it comes to the peas is that they're quite interesting that they can self-fertilize, meaning that a pea actually has both female and male genitalia. So they can technically self-procreate. They do not need to cross with someone else. They can, but there's no need. So this pea on its own can basically self-procreate from itself. So what he saw, basically, he took two tree breeding strains, meaning, let's imagine you have two different strains, one pea that is long and one that is short. True breeding means that if I take this long basically strain and it keeps self-breeding, it's always consistently going to give me long. Hence, that's what it means true breeding, that it's always going to give you that strain. And the same with the short, meaning that if the short keeps self-breeding, it's always going to give me short peas. So he took those two, and then he basically started, when he basically mixed them two, to basically cross them together, so the long and the short ones, he saw that the first generation of kids that basically came up resembled only one parent strain. So he took, let's say, a long and a short one. He crossed them, and he got only long peas. Okay, makes sense. This still makes sense with like, yeah, you're getting it from your parents. But then when he took these basically for the first generation, they were all long, and he basically now basically made them self fertilized themselves. Suddenly, you got the generation of the short peas again. But wait, now the first generation are all long peas. Now they're self fertilizing. Our second generation are long but also short. So what happened? But the parent was all long. Why did we are they now short again? So again, this kind of showed that it's not this thing of mixing just blood. It's that is not true. So then came Gregor Mendel. So I'm quite sure a lot of you guys have heard of Mendel. Mendel, we call him the father of genetics. Mendel has a very interesting story. Gregor Mendel actually comes from a very poor family. He's more of a peasant actually. Back in the day, they referred to him, more they kind of think of a peasants. He was basically actually grew up in a monastery, but quite relatively smart to the point that he actually during that time he managed to go to the University of Vienna to study mathematics. However, he did fail. He failed and he went back to his monastery. But in his monastery, still being very interested in science a lot, he did a lot of experiments of genetics that to this day we still use them. So what Gregor Mendel was using, he was using again this P, these literally P's that TA Knight that he was using. Why? Because peas were quite basically easy to manipulate. They're very, let's say, you can actually produce hybrids, first of all. So you can mix different traits to give you different other traits or other hybrids. So you can do a lot of experimentation with that. Pea varieties were available. You had many varieties. You had short peas, long peas, yellow peas, green peas, wrinkled ones, smooth ones, many with different types of varieties that you can then cross and create these hybrids and test genetics. Peas were quite small and very easy to grow. So that makes, of course, makes experiments very easy to be done with. And of course, they can be self-fertilized and cross-fertilized. So this is where you can actually really start testing a lot of genetics in some sort of way. So this is one thing that why pea plants were used a lot during genetics during those times because of its simplicity and of its let's say main characteristics that it had. So during Mendel's experiments, he literally did exactly the same as T.A. Knight. So his experiments will go in three stages. First of all, he would create basically a true breeding strain. So again, he would take a pea, for example, it would be he had a pea that was long. He would make sure that it was basically true breeding by he would make it self basically cross itself, so basically self-pollinate itself multiple times. And every time you would see that basically it would only produce long peas. So hence he made sure that they were true breeding. Then he would then cross-fertilize true breeding strains. So he would take a true breeding long pea and then basically a short true breeding strain and then make them basically cross-fertilize. And then he would basically get a trait. But one thing that I have to say that I have to commend Mendel for doing really, really, really well was that he actually was doing reciprocal crosses. So meaning that let's imagine in this case you have a white flowered plant and a purple flowered plant. So these are both, let's say, true breeding strains. So what he would do is he would take the pollen from the white flower plant and then fertilize the purple flower plant, but also vice versa. Because he said you never know. It could be that basically in some sort of way, if I only do white to purple flower pollination, there might be a skewing or something being happened. No, he basically made sure that this was equal. Then after that, you would get let's say the offspring coming up from basically the hybrids. So basically you would cross the self-breeding strains and then you would get the first offspring. And then he would allow them to self-fertilize for several generations. And then he would count the offsprings. So Mendel did exactly the same really as T.A. Knight. He really did the same. Got two true breeding strains, made them cross, got the children, and made basically the children self-cross. But why did basically Mendel get much more credit than for T.A. Knight? One very simple thing. Mendel actually counted. Mendel actually used mathematics because remember, he went actually to do mathematics in the University of Vienna. So he did have a lot of knowledge with mathematics. And then because of that mathematics, he started to develop a much more bigger theories than T.A. Knight. Just one simple thing. So again, this is basically, for example, if you look at the peas, this is how a pea looks like. You have this very beautiful, let's say, kind of pea structure, and then along with the flower, technically here. You have these basically these peas have these anthers. These anthers are basically these kind of structures that produce the pollen. So these are the ones that produce, let's say, the sperm. But within the pea, you have something called specifically the carpel. So this is basically the carpel, and the carpel technically contains the ovules. So what you can technically do is quite simple. You can, for example, let's say so let's imagine you want to cross these two together. So the purple pea and the white pea. So what you would do, you would take the purple pea, you would cut completely its anther so it would not be able to self-inseminate itself. So this one is not now able to self-procreate because you've removed now basically its sperm. You then go on to the basically white flower. What you do, you take a brush, you remove the pollen with basically a brush, and then you basically transfer the pollen to this purple pea again that you removed its anthers. And that's it. That's how you can do this cross fertilization. It's very easy and very quite nice. So what he did is basically he actually did monohybrid crosses. And what monohybrid crosses were basically he was studying two variations of a single trait, meaning that if you look basically at peas, peas can come in different varieties. They can come in with purple flowers. Sorry, they can have come with different colors of the flowers. They can come with different seed colors, seed textures, the pod color, the pod shape, position, and height. So in his monohybrid crosses, he would only take basically peas and he would only test one trait. So he would only look at the flower color. That's what a monohybrid cross is. So you would study two variations of a single trait. So that trait would be, for example, the color of the flower. So he would take two variations, either purple or white. That's what a monohybrid cross did. And of course, he knew he in his garden he had seven different traits, which is the ones that I just said right now. And he would cross them. He would cross them to see basically monohybrid crosses. So to the variations, either from the flower color, seed color, you name it. So each trait had two varieties always, and he tested them. So, as we said, what he would do is basically he would take these true breedings. So let's take an example of purple and white. He would take purple and white peas that were, again, true breeding, meaning that if they self-fertilize, they will only produce that one trait. That's what it means. When he made them basically cross together, so basically he would take then the first generate he would create the first generation. So he would get the purple peas, white peas, make them cross together, and then this would give you the first generation, which we call filial generation, F1. So F0 would be purple and white. This is F0. So these are your parents. So the children coming from the cross of purple and white, we would call them F1 generation, so filial generation. And what he saw was quite interesting. He saw that all of the F1 plants resembled one parent. They were all purple. So when he crossed purple and white, all of the kids were purple, all of them. And here he started to see, like, okay, we can see that one trait is dominant and the other one is recessive, meaning that the purple trait is dominant. It's way over dominant than the white trait. And the white is recessive, meaning that it's not going to be expressed if the purple one is there. And this really kind of proved a lot. Hey, how's this happening? Isn't there a mixing of blood? This is what we used to think. Why is it now the white blood not mixing? Nope, it that was not the correct thing. So this is where the experiment showed that what our thinking previously was not correct. So he then took basically again these F1 plants that were all, let's say, purple, and he basically let them self across. So they would basically self pollinate, and you get the second field generation. So the F1 generation now creates children, which now we call them the second filial generation, F2. And this is where he really got baffled because he used to think, okay, now that all of the F1 generation is purple, my second one should be all purple. No, he saw that now I have a 3-to-1 ratio, meaning for every four plants, three of them are purple and one is white. What happened there? And he did this again with all of these different types of traits, and he got the same thing. Every time he basically would get these basically true breeding phenotypes, he used to always see, okay, for example, with yellow and green, he would always get yellow in the first generation. And then if he got the first generation all yellow and let them self cross, they would become three of them yellow and one green. So he started to see like, okay, there's something, happening here. So he started to see, it's like, okay, three-fourths of the plant were dominant forms. So in our case, again, if we go back to the purple and white scenario, three-fourths were purple. And one-fourth was actually white. But if he actually got these F2 plants, this second generation plants, and he let them actually, let's say, self-cross, he started to see that this 3-1 ratio is actually a 1 to 2 to 1 ratio. 3 to 1 was actually buried, or let's say hidden here. Because what he saw, when he got these basically white recessive form plants, the 1 to 4th, if he made them self-cross, they would always give you recessive plants, meaning they will always give you white plants. So it was actually a true breeding white plant. But from this 3/4 of the plant, it was weird because 1/4 of those dominant forms of those purple basically peas, when he actually was able to cross them, were true breeding. Only 1/4 of them. The other 2/4, actually, no. They were giving us different varieties. They were not true breeding. That not every time they basically you cross them would give you purple. Some of them would give you white sometimes. So he saw that, okay, this 3/4 is actually one of it is true breeding that will always give me a breeding dominant plant. And the other 2/4 are not true breeding, meaning that they're purple. But if I self-cross them, they're not always going to give me purple. So he came up with this 1 to 2 to 1. And this was very interesting for him. So this is basically what happened. So all I said here, we can sum it up in this beautiful uh, graph. So he got basically in the first parent generation. So this is your F0. You have your purple parent, which is true breeding, meaning that if you self-cross this, will always give you purple. And you have your true breeding white. If you cross this, we'll always get white. So he crossed these together. So he did a cross fertilization. And the F1 generation, so the kid over here, was all purple. All of them. Interesting. So then basically, when he allowed this to self-cross, he got this 3-to-1 ratio that we were talking about. This 3 purple and 1 white. So this is your F2 generation. If he let his F2 generation self-cross, now giving you your F3, the white flowers will always give you white. So he's like, okay, this is true breeding. But in the purple flowers, only one out of the three would actually give you full, basically purple. So this was actually true breeding. But these two, not really. If I let them cross, they're going to give me sometimes white. So he saw, okay, it's an actually a 1 to 2 to 1. So one dominant true breeding, because we know the purple is a dominant color because of this here. Two non-true breeding, meaning that they're not true breeding. They're not just giving us purple, they might give us white. And one recessive breeding. So this was basically the 1 to 2 to 1 ratio that he was talking about. And this gave him a lot of different conclusions. First of all, we did not have intermediate traits, meaning that here we didn't have, for example, when we crossed this purple and white color, we didn't have, let's say, a mix of white and purple to give us, I don't know, maybe red or some other different color. No, there was no mixing, really. So this whole thing of blood mixing, that's not true. For each pair, for each pair of basically characteristics or traits, one trait was dominant and the other one was recessive. So in the case of basically here, purple and white, purple was dominant, while the other one was recessive. That's what he concluded. He also saw that pairs of alternative traits examined were segregated among progeny of particular crosses. So we'll get back to what he means here because this is a very important topic. But what he saw is that alternative traits were expressed in F2 generation in the ratio of 3/4 dominant to 1/4 recessive. That's what he saw. So this is basically in the F2, 3/4 were actually dominant and 1/4 was recessive. And he came up with his model. He said, parents transmit discrete factors, so different traits, as so we basically transmit traits as discrete factors, as one thing. And that one thing, he didn't know it yet, was actually genes. So he was correct. He just didn't know that genes existed during that time. That we are able to pass on a certain trait through one, let's say, one object, one discrete factor that turned out to be a gene. Each individual receives one copy of this factor, of this discrete factor, that now we know is a gene, from each parent. So this is why we might resemble our mom or our dad, because we get one copy of that trait from our mother and our father. Which one is more dominant? Which one is recessive? Depends. Not all copies of the genes are identical. If they were, we would basically look all the same. You have alternative forms of the gene, and that's called an allele. So we have a gene here. Let's imagine we have a gene that encodes for the color of the pea. That gene could be either white or purple. That's what we mean by an allele. Simple as that. An allele is a different form of the gene. We can be homozygous, meaning we have the same allele, meaning that, for example, in here, they have the same allele. They have purple, purple, most probably. They have here white, white. But this one most probably doesn't because it has to have some sort of white gene here to give this. So you can be either homozygous, meaning you have the gene that are the same alleles, or heterozygous, different alleles. For example, in the case you have a white gene and a purple gene. That's what we mean by homozygous and heterozygous. He also saw that alleles remain discrete. They do not blend, meaning that the white and the purple they does not blend together. They remain discrete. And the presence of an allele does not guarantee expression. It doesn't mean just because you have, let's say, the white allele means that you're going to have a white flower. No. Some are much more dominant and the other ones are recessive. We see that the dominant allele is expressed while the recessive can be hidden by the dominant allele. And this came up with two different things, genotype and phenotype. So, phenotype, that means your physical appearance, what you look like. So, in the case of the peas, the phenotype could be either purple or white. But the genotype could be different because we know that here, if you look at these ones two over here, this purple one, both of them have the same phenotype. This is purple, this is purple. They look the same. But genetically wise, they're different. The genotype is different because this one only has purple genes because all of the offsprings are all purple. This one has a white gene. So its genotype is different. So same phenotype, different genotype because this one can produce has the white gene that produces this white flower at the end. And then comes the principle of segregation, meaning that two alleles from a gene segregate during gamete formation and are rejoined at random, one from each parent during fertilization. So he started to see what's happening here is quite actually makes sense that because we know here, for example, more let's say the non-true breeding form here. So this purple dominant. This one has basically the gene of color and has two alleles. Most probably it has a purple allele and a white allele. And what happens is that they get separated during sperm formation or egg formation. They get separated. This is the principle of segregation. That during, literally, when you start producing offsprings, or basically, let's say your sperm or your eggs, these alleles that are together, there are two of them, two copies, they get separated, and you either give one of them. So in this case, you either give the white gene, or you give the white allele, or the purple allele. One of them. This is the principle of segregation. And then he was right. Mendel was so right because he didn't know about meiosis. And this is what meiosis is. I'm quite sure you guys have taken meiosis in high school. During meiosis, when we actually have the formation of an egg or of a sperm, our basically chromosomes get separated because we know we have two copies of a chromosome. They get separated. Well, each chromosome has actually a gene has a copy of that gene, as a copy, sorry, of the allele. So they get separated. So this principle of segregation is what happens during meiosis. And Mendel had no idea about chromosomes, about genes, about meiosis, and he came up with all this. So really, he had a very brilliant mind to think about this. So, of course, one thing that we can do, and I'm quite sure you guys have used this, is making a Punnett square. So what we're gonna do, what we like to do technically a lot in genetics, we like to use these squares. And where, for example, here we have these purple, let's say purple and white flowers, we know that the purple gene is dominant, so we put it as capital. We love to put it as capital. Here, this we resemble this would be the white one because it's actually very in a small case. This is just for us to know which gene, or sorry, which allele is, let's say, dominant and which one's recessive. So P for purple and capital. And this is little p basically means it's, recessive, most basically, and this is white. So what we do is that, for example, this is your father and this is your mother. Let's say, imagine again, we take this one over here. We take your non-true breeding purple flower and we basically let it self cross. So the sperm can give you either the purple gene or the white gene. And same thing goes with your eggs. Either gives you the purple or the white gene. And you do the square where you basically divide them and then again you do a cross, basically a cross together. So you bring this P over here, you bring this P over here. So this is like white white allele, white allele. So you're gonna get a white. So here you get the purple allele, white allele, you're gonna get purple. And on and on. You do this whole thing. And this is what you get. This three, basically this to 1 to 2 to 1 ratio. You have a generation that is fully genotyped BP, meaning that little p, so which is white. Then you have big P, small p, and the same thing goes here. So this is two. And then you have basically genotype that is capital P, capital P. So hence the 1 to 2 to 1. So this is the Punnett square. And you could use this a lot to do basically a lot, like to kind of, let's say, predict what the babies are gonna be, let's say what alleles they're gonna have, or let's say the ratios. So this is the Punnett square and how you analyze this. So now the question is, so quickly, I just want to see quickly the time. Yes, I have time for this. Perfect. Okay. Can we actually apply this? So this is all comes to plants. Can we apply this to human traits? Because, okay, it's great that you can use this for plants, amazing for, let's say, the crop industry, for the farming industry. That's amazing. But can you really put this genetics within a human being? And yeah, you can, because and I put this in bold and under cap, basically here underlined, some traits are controlled by a single gene. Not all, because some of them we exhibit a dominant and recessive inheritance, meaning that sometimes one trait, for example, that trait could be I'm giving an example, even though this is not true, but this is just an example, the color of our hair. We can say that our color of our hair can be dominated by one gene, either you're blonde or you're black, depending if you have the black gene or the blonde gene. This is not true because the color of our hair is multiple genes, but this is just to use an example. Some human traits can be controlled by a single gene, and we see a dominant and recessive inheritance. And this is very important, especially when we go more into genetic diseases. Because what we can do is we can sometimes track families to see if you have a genetic disease or not. So, give you an example here with glaucoma. So, glaucoma is basically a disease in where your optic nerve becomes completely degenerated, and this happens because of some sort of glucose deposition in your optic nerve, and this could lead to blindness. And it's mostly because you have a faulty allele. You have an allele that can really cause, let's say, this disease. So sometimes what we get is, of course, we have patients coming to the clinic, and they tell us that we have basically they're blind, and we start to see like, okay, do you know anyone in your family that was blind? And you start to see that, yes, there were a lot of people in his family that are blind. From here, you can start to see, okay, this is a genetic disease. And you know that blindness, genetically, there are a few probabilities. So you can actually even predict that this is glaucoma by just not even doing any examination, but just by asking questions. This is why in medicine it's super important, like if anyone goes to med school, like I remember when I was in med school, you would have like there was a huge emphasis on knowing how to ask the right questions to a patient. Because here, for example, let's imagine you take this female. So you have this pedigree, this tree. So this is your first generation, which they cross together, gives you the second generation. Then you get again the third generation from basically people getting married. So this means basically that they got married and produced kids. Same thing goes here. This is how you read a pedigree. So you have basically boxes, these kind of, let's say, little squares, which are males, and here basically circles, which are female. And you have basically unaffected, so people that do not have glaucoma, people that have glaucoma in green. So if we take this history, we can see that, hey, my grandparents, actually, my grandfather had glaucoma. And also, by the way, my dad had glaucoma. And he's like, okay, so your grandmother didn't have glaucoma and your mother didn't have glaucoma. That's interesting. Do you know anything else? Like, yeah, my aunt has glaucoma and both her children have glaucoma. If you really build this, you start to see like, hey, this is generally quite dominant. Like this allele is quite dominant because every single time, because in this case over here, if this was recessive, none of the kids would be actually having glaucoma because the if basically let's say the gene that causes glaucoma was recessive, why would any of them have it? Because they're gonna get a good gene from their mother. So this can tell us a lot that this is actually a dominant gene. And then gives you, for example, albinism. Albinism is basically a form that you have you're you don't have in some sort of way melanin or a pigment. So you have a lot of basically these kind of spots that are really white because it's literally in some sort of way what happens here. You have an allele that gives you a non-functional enzyme called tyranase, which is needed for pigment formation. And again, here you start to see that, okay, you have this basically first generation, your father and your mother that are crossing, and you have all of these basically kids that do not have albinism. They don't. But then suddenly in the third generation, we have it appear. This tells you a lot that you have a recessive trait because it always skips. So a generation. So the first generation that you do not have, then the second generation, you should not have it if it's recessive. If the first generation, none of the parents have the disease, and then your second generation doesn't have it, but then your third does, it means it's recessive. Because here you do have it. It doesn't skip, it always is present in a generation. If it skips a generation, it means that it's recessive. This is how sometimes you can actually see if something is dominant or recessive. So quickly see the time. Yes. So this is the last thing I will be talking about. So what Mendel also did is that, okay, he found that this is great, this is amazing. I got to see all these things. Let me do a dihybrid cross, meaning that now, rather than just basically testing one single trait, so meaning the color of the flower, let's do two different actually traits. So what he got two breeding lines of two traits. So he got peas that were round and yellow. So he knew that the round was dominant over the wrinkled. So that's why he put this as small little r. So you basically have the trait, so one gene that encodes for the shape of the pea, if it's round or if it's wrinkled. And then he got the color. Basically, he got these were round and yellow peas. These were round and green peas because he saw that the yellow allele is dominant over the green one. And he crossed them. And he saw that in his first generation, just like in the other experiments, had basically this kind of one parent, all showing just the dominant phenotype, which were they were all round and yellow, all of them. But then when he allowed these to basically self-fertilize, so giving you now the F2 generation was actually really cool. He got this 9 to 3 to 3 to 1 ratio, meaning that. So let's take this and actually put this in a nice kind of cross. So you have here these are your round and yellow peas. These are your green and wrinkled peas. So what he did is basically like doing meiosis. You get basically one copy of each. So you get a copy For example, let's say in this case, you have the round gene and then the yellow gene. Here you get basically the wrinkled gene and the green gene. You let them cross-fertilize, you get the first one generation, which is purely, completely all round and yellow. But this is very different to this because we know that this is heterozygous, meaning that they have basically different genes, sorry, different alleles. My bad. And then you allow this to create this meiosis. Sorry, not meiosis, but more, let's say, self cross. And you basically get this following. So you have 9 out of 6 are yellow, 3 out of 16 is round and green, 3 out of 16 is wrinkled and yellow, and 1 is out of 16 is wrinkled and green. So this was basically what he saw. So technically this 9 to 3 to 3 to 1 is technically just the addition of technically the 1 to 2 to 1, if you think about it. It's what he saw. But what this really told him is that, okay, in this dihybrid cross, the alleles of each gene gets assorted independently. So he saw that genes do not are not influencing each other's way of being distributed. They do not. So this gene and this gene basically they do not basically interfere with each other. That's what the principle of independent assortment meant, meaning that a gene, or basically let's say something of a gene allele, the alleles will independently assort compared to a pair of other alleles. And what happens during metaphase one. So if you go back in here, so you have basically this gene over here, which basically you have two alleles. You either get a blue or a blue basically one, and here the yellow. But this one doesn't affect this one. These get this and this independently are assorted. So you either get this one or this one. So you get this. And from this and this, you get one of these. So for example, in this case over here, you either get a round gene or a wrinkled gene. Or you get basically a what do you call it? A yellow or a green. But they don't affect each other because this assortment, so when you divide these two, you either pick this or this. And the same thing with it. They're independent of each other. That's what I'm trying to say. Hence, it comes of the principle of independent assortment, that genes assort independently of each other. And that's what Mendel proposed, and it's true. This is exactly what happens during meiosis. Homologous chromosomes during the metaphase one will segregate homologous chromosomes. So basically different chromosomes, they do not pair up. Okay, guys, this is the end of today. Is there anything that you want me to repeat, to go over? Please let me know if you have any questions.
No, thank you, doctor.
All right. Thank you so much, guys. Hope you have a nice continuation of the week. If you guys have any questions or anything, please, please, please do let me know. Have a great one, guys. Thank you so much.
Thank you, doctor.
Thank you.
Thank you.